In the heart of the lush rainforests of Central America, a miniature jewel thrives, the Oophaga Pumilio, commonly known as the strawberry poison dart frog. Join us on a captivating journey into the intricate world of this tiny amphibian, exploring its evolutionary history, ecological significance, and the challenges it confronts. Among Earth's most toxic yet brilliantly colored creatures, the strawberry poison dart frog exemplifies the dichotomy of danger mingled with beauty characteristic of life's elaborate diversity emerging from forces of creativity and destruction. As a species, the amphibian Oophaga Pumilio mirrors such contrast in its role within tropical rainforests, at once playing keystone participant across cascading food chains yet fragilely dependent upon intensely specific conditions forging balance amidst chaos. In both appearance and ecology, Oophaga Pumilio embodies the delicate interconnectivity allowing ecological splendor and services to persist. But severe risks now jeopardize the stability that enabled their niche, habitat and biome itself to thrive to the modern day. Their fate thus serves as a microcosm for tropical wilderness teetering at extremes capable of collapse through mankind's strained relationship with wilderness itself. Evolutionary history. Like over 300 related dendrobatid frog species populating neotropical America, the lineage giving rise to modern Oophaga took hold around 50 million years ago during the Eocene epoch as South America drifted northwards while climates grew warmer and wetter, ideal for diversification. Through the Miocene era, proto-poison dart lineages then expanded outward, tracking the blossoming of rainforest ecosystems where few predators could endure the toxic alkaloid compounds seeping from their skin as chemical defenses. Over successive periods of forest contraction and regrowth driven by Andean tectonics and cycling climates, genetic isolation carved variation eventually designating modern genera including Oophaga emerging roughly 5 million years ago along the Isthmus of Panama land bridge. Recent evidence suggests Oophaga Pumilio itself arose through allopatric speciation processes as little as 800,000 years ago when Pleistocene forest fragmentation created genetic distinctions between populations. In location lending its namesake, the San Jose region of eastern Panama and adjoining northwest Colombia sustained founders of what became the strawberry color morphs, one among over 15 distinct varieties now identified across islands and mainland rainforest tracts. Yet whether such unique diversity actually constitutes separate subspecies or microspecies remains debated within the scientific literature. Adult Oophaga Pumilio exhibit physical features characteristic of most dendrobatids including robust bodies measuring roughly 1.5 inches between nose and vent aided by expanded finger and toe pads granting superb climbing ability integral to avoiding terrestrial predators. Smooth skin colored by 3-layer dermal pigment cells hosts alkaloid toxins as chemical defense representing some of the most deadly secretions in the animal kingdom capable of causing respiratory failure in potential attackers with just microdoses. Sensory adaptations like the inner ear papilla grant expanded high-frequency hearing enabling complex vocal communication including courtship signals and territorial warnings. Large protruding eyes enhanced by spectrally diverse cone cells boost vivid daytime vision critical navigating rainforest floors. The most obvious trait remains the strawberry phenotype's vibrant scarlet dorsal surface bordered in royal blue legs and azure feet, a distinctive example of aposematism where vivid coloration denotes toxicity avoiding misidentification by common predators. But beyond the 15 classified color patterns spanning every morph known to science, an incredible 800 further nondescript varieties recently emerged exhibiting muted grays and greens likely signaling transitional specimens undergoing selective pressures. Distinguishing such individuals remains key for tracking microevolution pathways in action. Ecology. Strawberry poison frogs fulfill keystone roles across rainforest floors as mid-level consumers feeding on small arthropods like ants and isopods yet comprising critical prey for snakes and birds that carefully handle these toxic tidbits. By regulating invertebrate populations that would otherwise unchecked spread disease or dominate plant-insect foundations underpinning all terrestrial tropics, Oophaga Pumilio secures ecosystem balance. But they additionally serve direct plant allies, as tadpoles hatched from terrestrial eggs laid across tropical leaves soon wriggle their way towards protected pools of water trapped by foliage in bromeliads, rain-filled plants that cannot complete reproductive life cycles without frog transport of key nutrients when they die and decay. These interdependencies stabilize the ecological networks allowing forests to sustain such opulence. Yet narrow thermal and moisture niches restrict all dendrobatids. As diurnal ectotherms closely regulating metabolism through ambient conditions, minute alterations beyond 20 to 30 C or 50 to 100% relative humidity swiftly impact health and population numbers unable to escape local microclimates within terrain-bound habitats. Current models suggest upwards of 30% range losses may occur for Oophaga Pumilio over coming decades as climate zones accelerate beyond envelopes that long maintained balance through the Holocene to shelter anurans here over 10,000 years. Habitat and behavior. Inhabiting leaf litter floors up to 100 meters in elevation within lowland and mountain rainforests receiving ample annual precipitation, Oophaga Pumilio roams loose territories up to 20 square meters daily consuming insects and arthropods located using enhanced color vision. Hyperlinkage between regional climate patterns and restricted dispersal renders local groups profoundly vulnerable to extreme weather shifts and habitat quality decline still within survivable ranges but exceeding optimum conditions this species long relied upon through generational timescales. During mornings when cloud forests mist, these diurnal amphibians emerge from moist hiding spots below epiphytic plants, logs and leaf piles used overnight for concealment from army ants, spiders and snakes which disregard their toxicity. Now raising alerts if threats approach, they exploit a few daylight hours to actively forage centipedes, mites and termites using extendable tongues before heat or humidity compels seeking cooler shelters by early afternoons. Here courtship rituals commence when ready males vocalize buzzing, clicking and trilling calls that females amplify by selecting responsive mates after approaching their protected sites. External fertilization occurs involving the deposition of pigmented gelatin egg clutch masses conferred antimicrobial protection while embedded with ordinarily harmless bacteria that soon biologically re-engineer the jelly matrix into potent toxins protecting vulnerable developing tadpoles over 2-week incubation and week-long carry to canopied aquatic nurseries housing them until froglets finally morph. Conclusion. While Oophaga Pumilio demonstrates resilience settling new forest patches, climate change progresses beyond conditions enabling persistence even across scattered refugia colonies inhabit. As fragile stability unravels for communities fine-tuned through deep time, once harmonized networks now fracture past points permitting recovery within human lifetimes alone. While no threat extinction now endangers Oophaga Pumilio outright according to prevailing assessments, localized declines signal growing instability as most brilliant species often disappear well before appreciated at valued more than mercurial economic priorities alone. But incentives still exist to enact conservation efforts from habitat corridors to captive breeding assurance should outcomes continue deteriorating faster than institutions adapt. However, without addressing root factors degrading wilderness itself, any last-ditch efforts cannot restore balance eroding beyond sustainability. Life finds pathways even emerging more spectacular perhaps in some distant future. But such transformations now unfolding assure to be long, chaotic and less familiar than worlds nurturing rainbow biodiversity filling nature's palette through eons of shared creativity that now demand defense so not to teeter towards becoming but memories alone through dissolution of relationships that gave vibrant meaning to empty expanses glimpsing possibility since vanished in silence. Choices remain as how to orchestrate closure to threatened biomes or see their reprise towards equilibriums where life and land again infuse one another.